Hi, we are students from Harmony Science Academy. The science behind our experiment is the process of our digestive system and how it breaks down different foods containing lipids, carbohydrates, and proteins using certain enzymes. People eat food every day in order to convert it into essential nutrients such as peptides, amino acids, fatty acids, and glucose. How the body does this and the scientific process and principles behind it is our driving question and the main purpose of our experiment. We hypothesize that certain enzymes break down specific molecules depending on what the food contains. Looking more in depth into the experiment, the reason we specifically looked at the processes of pepsin, amylase, and lipase is because amylase initially breaks down starch in the mouth into carbohydrates. Then pepsin breaks down protein into amino acids in an acid environment, the stomach. Finally, lipase catalyzes the hydrolysis of milk into fatty acids in the small intestine. Our ELA component is a series of journal entries dictating what we did each day throughout the PBL experiment. For more info on what we specifically did each day, please refer to our website.
What we're talking about here is how the history of modern enzyme technology began in 1874. Before this, a lengthy evolution had occurred where enzymes were being used for many different purposes. For more information, please visit our PBL website. For the materials, we used many different types of solutions, as well as test tubes and a water bath. For procedure 1, we used a pipette to put only protein solution into tube 1 while we added protein solution and pepsin solution to tube 2. Then we placed in the water bath for 15 minutes and observed the color change. For procedure 2, we used a pipette to put only litmus milk solution into tube 1 while we added litmus milk solution and lipase solution to tube 4 and then observed the color change after the passing of 4 minutes. For procedure 3, we added starch solution to tubes 5, 6, and 7, amylase solution to 6 and 7, and amino solution to 8. Then we added iodine solution to 5 and 6 to test for starch. Then we added Benedictine's reagent to 7 and 8 to test for glucose. Then we recorded the color change.
In conclusion, proteins, pepsins, lipids, and starches have been proven to be digested by enzymes based on their reactions in the experiment. The color they changed to proves that there was an enzyme present in order to digest it. Proteins change color with polypeptides and amino acids. Lipid solution changed based on acidity, and starch changed based on the presence of glucose.
For more info, please refer to our website. Thanks for watching.